hour number 7, amazing. And Dr. Furbus, I think you're online. One of the reasons we set up this 24-hour AIDS research marathon was to make sure the general public learned more. And I can see already we have at least the next generation. I think one of your children are on saying, "Go mom." I love that, because I think for all of us who work in this field, this is a family affair. We don't do this on our own. We do it with the support of our family and our friends, all of us. And I am delighted to hand off the hosting baton for hour 7 to Sophia Zamudio-Haas to take us into the next hour. Welcome.
Thank you so much. Pleasure to be here with you all and hosting. Hello, good afternoon from Berkeley, California. It's my pleasure to be hosting this next hour, the 7th hour of our 24-hour live stream to save HIV research. I am actually right now at a 2-day convening for California HIV/AIDS research programs, and really just glad to be here today. And we've got an excellent lineup for you all over the next hour. And yeah, just such an important topic, such a timely moment to be doing this and sharing experiences of researchers, community members, and really showing about how life-saving and critical these efforts are. So with that, I am really happy to be breaking in this 6th hour, with Dr. Connie Celum, who's a professor of global health and medicine and the director of international clinical research and the CFAR at the University of Washington. She is going to be presenting on HIV and STIs, intersecting epidemics, novel interventions.
Thank you so much, Sophia. Am I coming through okay?
Yeah, sound great.
Well, I'm calling in from Johannesburg, South Africa, and so if I look a little blurry-eyed, I am, just a little bit jet-lagged, but it's such a pleasure to be here, and I'm really glad that I was able to catch the video of Kathy Furbus, because I think it is always helpful to someone I'm getting to know who does a lot of TikTok things told me recently, he's a scientist, and he said what people want to know is that scientists are humans. So I will tell you, my background is, before I get into the meat of the talk, is, I grew up in a very conservative part of California, and when I went to UC San Francisco in 1980 to be a medical student, the first most impactful experience I had on the wards was Paul Volverding running down the hall with a typed out manuscript about Kaposi sarcoma. And we were really, it felt like we were on the front lines of what felt like just a unbelievable moment in time, where Kathy talked about this, seeing people, in my case, I was in my 20s, and I was seeing people younger than me have 3 days basically to come to terms with their death, often coming out to their family. And we had very little to offer. We didn't know what we were doing. So it was like palliative care, and that's not what I thought I was going into. So I really made a commitment to myself. When I can still honestly remember many of those first men who I took care of, because it was at that point primarily men who were dying of AIDS. And I said, what can I do? And I made a commitment, because we didn't know what the virus was. We didn't know it was a virus. And I made a commitment to do prevention. And so that's been my life work, and still gives me a lot of passion. And I have to say, gives me hope. But also the last 6 months has given me a sense of frustration, because we have tools and we cannot afford to not use them. So with that, I will jump into the topic and see if I can maneuver this. So I'm going to talk about
Hi, all. Just bear with us for a moment here while we get this worked out. Dr. Celum is just going to be sharing her screen. And I'll take a moment here just to say that actually I didn't really introduce much about myself, but I'm assistant professor at UCSF, and I first met Dr. Celum when I was a HPTN domestic scholar for her completed HPTN 082 Vanguard trial around oral PrEP with young women and adolescent girls in South Africa and Zimbabwe. So much respect. Thank you.
Thank you. Well, I just learned there's one button not to hit, which exits you from the program. So try to not do that again. But I will talk about STIs and HIV because when I made the decision to go into prevention, I was very interested both in women's health and STIs and reproductive health and HIV prevention. And somehow in the end, it all came together, but it wasn't always clear exactly how. Let's see if I can advance my slides. I'm really afraid to touch that button now.
Hi,
Jeff?
Connie. It's Jeff here from the backstage.
Yeah,
I think
where's
we're
the
going to have
button?
to upload your slides again, Connie. I'm so sorry.
Okay.
But we
Sorry.
can get that done. We'll work with you to get that done.
Okay.
I'm
Sorry.
going to send you to the backstage for a second. And
Well, they're
there
there.
we are. They're ready.
Okay. Phew. Okay. Sorry. Some of us who are old now believe in IT gremlins, and they chase me all over the world. So you just got to witness that they followed me to Johannesburg. But without further ado, I'll just jump into the talk, and I will tell you why I've been fascinated by the intersection between HIV and STIs and tell you some of the things that we're learning that have led to interventions. First of all, to put it in perspective, we have an ongoing STI epidemic, and we have had much less attention, honestly, much less advocacy focusing on STIs. And that's partly why I've tried to straddle this area, because it feels like, there is still so much stigma associated with STIs. In my role as a clinician, the two things that could bring people to tears was telling them they had herpes or HIV. Now, HIV, people take it often with a pretty sanguine response, but STIs are, especially when you have to live with it for a lifetime like herpes, and for which we don't have great interventions, is still a real problem. So the key things I want people to take away are that we have ongoing high rates of chlamydia, gonorrhea, and syphilis, and now, to my great chagrin, we have congenital syphilis. And I'll try to break these down a little bit without going too much into the weeds. But I think first, it's just really important to remember that when we talk about STIs, actually, for most people who acquire HIV, it is an STI. They acquire it through sexual transmission. And it is a synergistic epidemic in the sense that the populations who are at risk of HIV and STIs share a lot of things in common. And then lastly, there are a lot of data, epidemiologic data primarily, that suggests STIs are cofactors for HIV transmission. I'm a very pragmatic person, so when I started seeing these data fresh out of medical school and residency, as we learned more about HIV, I was convinced that we should be able to use this to develop interventions. And the data, as they evolved in the 80s and 90s, indicated that genital herpes, usually caused by a virus called HSV2, which is a lifelong recurrent episodic infection, puts people at risk by about threefold because it causes little ulcers, sometimes not symptomatic, often not symptomatic, and it becomes a portal of entry for HIV. Syphilis also causes ulcers, tend to be even less symptomatic, often are really not noticed, and that increases people's risk about twofold. Even a parasite called Trichomonas vaginalis increases the risk of HIV. And then human papillomavirus, which is associated with cervical cancer, anal rectal cancer, laryngeal cancer, increases the risk about twofold. So one would think that we should be able to use this information to develop interventions. But I want to pause for a moment because I think I said it, but I want people to really hear it. This is really a jaw-dropping curve that we in the 20 20s are seeing rates of syphilis that are comparable to what we're seeing after World War II. And we have antibiotics. We have very effective antibiotics for syphilis. We're seeing it through that slope is a little steeper in the West, but we're seeing it in all regions of the US. The rates are not just high in men who have sex with men, but we're beginning to see it in men who have sex with women and women who have sex with men. And what that has translated to is about a over fourfold increase in congenital syphilis, which can lead to infant death, stillbirth, and a number of very serious sequelae. So why is that happening? We should have the ability to diagnose and treat it. But part of it is we're not doing well in terms of populations who need prenatal care or who have a diagnosis, but then we can't get them in for treatment. So honestly, it's a failure of our medical care system as much as our public health interventions that we're seeing these very high rates of congenital syphilis. So you might ask, well, why? Why is this happening? And it is because of health disparities. If you look at who's getting these various STIs, there are higher rates in certain parts of the US. There are higher rates in people who don't have health insurance and in states that didn't have Medicaid expansion. It's also happening because of stigma. And anyone that I can think of so many cases where when I had to give someone a diagnosis of an STI, the next question is, where did I get it? And then secondly, how do I tell my partner? And then lastly, social determinants of health are real. And that people who don't have insurance, who are not able to take time off if they do work to get into healthcare, or have mistrust of the healthcare system are all reasons. Lastly, we have been slowly dismantling our public healthcare system. And that got worse during COVID, where many clinics had to pivot to try to respond to COVID, and STI services became much more sparse. Provider awareness. I have seen a fair amount of syphilis and gonorrhea, chlamydia, but many primary care physicians have not. And if you don't ask, you won't find. If you don't look, you won't find. And so these are mainly asymptomatic infections. So the patient may not come in saying they have something, and the provider may not diagnose it. These are largely asymptomatic infections. And our diagnostics, some of them are pretty archaic, like we're using a serologic test for syphilis that we've had for decades. And it is not by any means state of the art. We're seeing antimicrobial resistance, particularly in gonorrhea, and then another bacteria called Mycoplasma genitalium. And then we have tools. We have vaccines that prevent human papillomavirus, hepatitis A, hepatitis B, and we don't use them enough. And then there's also an intersection between substance use, HIV, and STI. So there are a number of reasons why this is happening. But it's not all doom and gloom. Part of it is also that we've had some successes. We now, as you're, I'm sure, hearing on this marathon, we have fantastic tools to treat HIV so that people can take with a pill a day, or in some cases injections, they can become undetectable. Doesn't mean they're cured of HIV, but they're not infectious. So that campaign became known as undetectable equals untransmittable. It gave people living with HIV yet another motivation to take meds. But it also meant that condom use has gone down. Around the same time, we started developing effective ways for primary prevention for someone who's HIV uninfected to remain HIV infected. And the people who need PrEP are often people who've had STIs. So there's been a lot of literature written about this. It's not that PrEP caused STIs, but if we're doing a good job at targeting PrEP to people who most need it, it is people who have STIs. So I think really in the last, 5 to 10 years, I think we've gotten serious about the fact that the approaches we were taking about talk about it with your partners, get tested, get treated, those are all very solid pillars of responding to STIs, but they're not enough. And so there was efforts to try to help providers do a better job at opening up in a non-judgmental way conversations about whether they should be thinking about STIs. But honestly, we don't do a very good job at that, and I include myself in that way. It takes time to touch on a sensitive topic, and when you have 10 minutes per patient, guess what often doesn't happen is open-ended conversations about sex. We also, as I just mentioned, haven't done very well in using vaccination. We are doing better, but still have a long ways to go in scaling up and implementing HIV PrEP and post-exposure prophylaxis. We need to discuss STI prevention in the context of contraceptive services, including emergency contraception. That's a population who might also benefit from PEP. We need to do better at our clinical delivery. If you don't see a lot of STIs in your primary care practice, you may be using antibiotics that are no longer recommended. Another area where we have a lot more work we could do better is partner services. And that is challenging because of the stigma that still exists with STIs. It's not easy to tell someone that you've been diagnosed with gonorrhea, chlamydia, or syphilis, and that they should get tested. Just going to dive into a few areas where I want to highlight how moving from epidemiology and understanding how prevalent STIs are, who gets them, etc., does not always end up being a linear course to identifying interventions. And I'm going to collapse about 7 years of my career into a few slides and tell you that HSV2, we thought that is going to be the STI that has the biggest impact. Highly prevalent. In some populations, 1 out of 5 people without HIV have it. Up to 80% of people with HIV could have it. Often asymptomatic. We have some good serologic tests, and we had over 30 studies from around the world that suggested it increased the risk of HIV by 2 to 3-fold. And some math modeling suggested it might account for as many as a third of incident HIV infections in Africa. So I started doing global health research when I saw these maps and thought, we should really find out we don't have a vaccine for herpes, so let's see if we can suppress it and reduce the risk of HIV. So we had 2 hypotheses, and this was based on many, many studies. And to the extent that some ethics committee said it's not ethical to randomize someone to acyclovir or placebo, you should just be giving everyone acyclovir. But honestly, we didn't know if suppressing herpes would reduce HIV susceptibility. So this was my first ever international multi-site study that I co-led with a number of colleagues, where we enrolled persons who didn't have HIV but did have HSV2, and about almost half were cisgender women, a little over half were men who have sex with men, and it was a very simple design. They had HSV2, and we would randomize them to standard acyclovir twice a day or placebo, matching placebo twice a day, and then see whether we could reduce HIV. On the right, we showed that the 2 curves, in pink, the pinkish color is placebo, and blue acyclovir, they totally overlapped. It did not work. And this was in about 2007 when we got these data. And it really pointed to me, the lesson here was, I still don't think our hypothesis was wrong, but we don't have good enough herpes suppressive drugs to really totally ameliorate the inflammation caused by herpes. So it's still an important cofactor. We still need a vaccine. We need better drugs. In parallel with that study, which was funded by the NIH, the Gates Foundation funded what we called the sister study and said, well, what about herpes suppression for people who had both HIV and HSV2? And we took this study on in about 2008. And we didn't, at the time, it was by no means the standard of care to offer HIV treatment to everyone, because we had didn't have the simple and highly effective treatments we had now. So people would get treated for HIV when their CD4 counts dropped. So it was ethical at that time to randomize people who were not yet eligible for antiretroviral therapy following national guidelines. And this study was done in 14 sites in 7 countries in East and Southern Africa. And we did the same regimen, twice a day acyclovir versus a matching placebo. Harder study because we had to enroll both the person living with HIV and their partner. And we followed them for 2 years. And this was pretty devastating to many people because, again, it was flat. But we did lower HIV viral load, but just not to the extent that would translate to a significant reduction in HIV transmission. So those 2 studies were hard ones to do, the first ones, because it felt like it sort of put STIs on the back burner for a while. If we couldn't impact HSV2, what were the other priorities? So it felt like kind of STIs stayed on the back burner for almost a decade. And I want to, sorry, you were going to
Sorry, Connie, just giving you a moment just to say, it's so hard to interrupt here. I love this presentation. I think we've just got a few more minutes here in case there's some comments or.
Well, I just want to say, don't.
Yeah, thank you.
I'm just going to touch on real quickly bacterial STIs because I think we do have more progress to report there. First of all, they're incredibly common. You're going to hear more about PrEP from my colleague, Dr. Raphael Landovitz. Incredibly common. I've done studies in Africa where 1 out of 3 women have gonorrhea, chlamydia, or syphilis, and there are high rates in men who have sex with men. And these just show you some of the numbers where it's not just an entry, but over a year, 1 out of 3 in many cases will acquire an STI. So what can we do about it? And I'll wrap up by just saying that we did a trial in San Francisco and Seattle that asked the simple question of, could antimicrobial post-exposure prophylaxis reduce STIs among men who have sex with men? And the answer was yes, we could reduce it by 2/3 and without having really any significant change in sexual behavior. People were really motivated. They wanted to reduce their risk and their partner's risk. We did an open label extension. We saw that persisted after we announced to the study participants that it worked. So that was also encouraging. There have been 3 studies now in men who have sex with men that found very similar reductions. It's particularly effective for chlamydia and syphilis, less effective for gonorrhea because of existing antimicrobial prophylaxis. And we now have CDC guidelines. So research can lead to public health interventions. It can also lead in a very short amount of time to public health impacts where we're seeing reductions in gonorrhea, less so in gonorrhea, but in substantial reductions in chlamydia and syphilis. These data are from San Francisco, but this is being reported in many parts of the world. And the next chapter, and stay tuned, is we are now working on vaccines, including asking the question of whether a vaccine that protects against meningococcal B, which is very closely related to gonorrhea, can protect against GC. So a year from now, we'll have answers from those studies. I just want to end and just say that there's so much work to be done in STIs globally, as well as in the US. And I think there's some very, very exciting work that's happening, and we're already beginning to see some results with like DoxyPEP. Thank you.
Oh, thank you so much. Thank you so much, Dr. Celum. That was really fantastic. Just so many important points there. Just really great to see that all laid out, both, you know, previously and then up until right now, and really looking at the ways that you can't really separate out one from another, as you were pointing out with the COVID impact on STI, really interconnected health considerations around HIV. So really lovely talk. Thank you so much.
Welcome.
Lovely. Okay, great. Well, we're now going to transition to our next speaker for this hour. And again, thank you so much to Dr. Connie Celum and sharing that beautiful summary around intersecting epidemics with HIV and STIs. I'm going to bring in now Dr. Sunil Suhas Solomon. My pleasure to introduce this professor and vice chair for research in the Department of Medicine at Johns Hopkins University. He will be presenting on heroin to health, global drugs, global diseases, and global solutions. Thank you so much. Welcome.
Thank you, Sophia. Perfect. I wanted to start by thanking the organizers for the opportunity to present some of our work here today. It truly is an honor. And thank you all for logging in wherever in the world you are. I want to just start with a little bit about myself. I finished my medical training in India and I came to Baltimore to pursue a master's in public health in 2003. So from a sheltered life in India to being an immigrant on the streets of Baltimore, and more importantly, a student at the School of Public Health, drug use was suddenly front and center in my life. In 2004, heroin was probably the most commonly used drug in Baltimore, and people who injected drugs had a disproportionately higher burden of HIV and viral hepatitis compared with the general population. But over the next 10 to 15 years, we saw a decline in the number of new infections among people who inject drugs in the Baltimore and the US, so much so that it sort of lost attention. It was no longer on the front page of like every newspaper. But then there was this resurgence of drug use all over the US, even in places where drug use wasn't traditionally endemic. And this was really fueled by all the synthetic drugs, and the number of people succumbing to overdoses in the US reached new heights, with over 100,000 lives lost in 2023. So I run down this alley in Baltimore County every morning, and I often see injection paraphernalia lying all over the alley. So what was initially a problem in Baltimore City was now spreading into the county. And every time I drive home after my run, I see the sign of the number of overdoses and lives lost gradually ticking upwards with each passing day. I think it's very important for us to remember that each of these numbers represents someone's mother or someone's father or someone's son or someone's daughter. And for those of you who are not familiar with substance use, the increase in overdoses generally tends to be the first sign. So this is usually followed by outbreaks of hepatitis C, followed by outbreaks of HIV. In fact, we've already started seeing outbreaks of HIV among people who inject drugs in the US in places where we've never seen them before, like in Lowell County in Massachusetts, or across all the Appalachian states. But drug use really is a global issue. It's estimated there are about 15 million people who inject drugs globally. That's about 0.2% of the global population. But this 0.2% accounts for about 5.5% of all people with HIV across the world, translating to a burden that's almost 27 times higher than what it should be. And if you look at hepatitis C, which is more transmissible by blood than HIV, people who inject drugs bear a burden that's almost 60 times higher than what it should be, really highlighting the vulnerabilities this population faces. And really that was part of the reason that drew me to work with this population, because everyone told me it was very hard to do. But South and Southeast Asia account for about 33% of all the people who inject drugs globally. And so I decided to go back to India and start my work there in 2004. So we set up a research site in North Chennai, which is all the way in that orange state in the bottom. And because North Chennai was the part of Chennai where people who injected drugs resided. We replicated some of the work from Baltimore in India, and we found that there was a lot of HIV and a lot of hepatitis. But when I referred them to our clinic, which was in South Chennai, most of them would not go. They would cite distance as a factor. So we actually got medications donated from pharmaceutical companies, and we set up a clinic at that research site. I used to go there every Saturday morning to see patients who were showing up. We provided them with free care for several conditions, except for antiretroviral therapy, because that was being provided free by the government of India, and it's a lifelong commitment. So I used to refer people there, and I still remember waking up on January 1st, I think it was 2005 or 2006, to a text message that one of my first patients whom I had seen at that clinic had died of AIDS. And this was just not acceptable. 1 in 3 people with HIV was dying of AIDS despite free medication available. So I talked to a bunch of my patients and I asked them, what's the problem? It's like, free medications is just about 20 minutes down the road. Why aren't you going there? And they told me like they are daily wage earners. So they need that day's wages for their food and for everything else in the house. And money to them was more important than HIV care. So what was the point of going there? So I asked them, what if I actually paid you these wages you lost because you went to the clinic to pick up your medicine? And this answer led us to designing one of the first trials to evaluate the role of cash transfers in improving HIV uptake among people who inject drugs. So in this figure, the higher the line, the better. So the dark green line that looks higher means like it's a very positive outcome. So essentially what we found was people who were assigned to the cash transfer are almost 3 times more likely to start treatment compared to the control condition. And these cash transfers could be redeemed for food or for other essential items. So in many ways, it benefited the entire family and not just the participant. So this entire trial cost us about $100,000 to do in India. But the findings from this trial have now been replicated in multiple studies across the US and even with other populations. For example, they're looking at, can we use cash transfers to keep women in school in Africa to prevent HIV infection? Can we use cash transfers to improve vaccine uptake across Asia? So though this trial was designed as something to look at HIV outcomes among people in Chennai, it had global implications. But Chennai was just one city, and I'm someone who always wants challenges, and so I wanted to go even bigger. So we expanded our work across India, and at the time we started this work was about 2012, and there was a lot of discussion about the drug use epidemic in Punjab, which is and like this New York Times article, which actually compared the epidemic in Punjab to something we would see in New York or in Baltimore, which had originally motivated my work. And we quickly found that the HIV epidemic in these places outside of Chennai was several orders of magnitude higher than what is being seen anywhere in the world. So just to give you context, that big red circle with the 12.4 was the city of Kanpur. So that 12.4 essentially means that if you took 100 people who injected drugs who were HIV negative, by the end of 1 year, 12 of them had picked up HIV infection. To compare that to what it was in Baltimore in the same time frame, it was about 1 in 100 or probably closer to 1 in 200. So really showing you that huge disparity in incidence between these two regions. So for the past decade, our group has been trying to address this high incidence. The first thing we recognized was that services to prevent and treat HIV were available, but not accessible. The problem was that people had to go to one place for their methadone, another place for their syringes, another place for testing, another place for their ART, a different place for tuberculosis, a different place for the liver. Like, I'm getting tired just reading it out. So you can imagine how patients or clients felt at that point in time. They were people, right? And people like us. And so we have almost evolved to a society where we want Uber or Lyft or Grubhub or whatever it is in your region of the world to bring stuff to us, that it was practically impossible to expect people to go to all these places, but that was the standard of care in 2012. So we asked the question of, if we brought all these services under one roof, would it improve HIV testing? To most of you, it may sound like a complete no-brainer, but at 2012, there was no data from any trial that supported this. And unless you have data from trial, it's very hard to move that policy needle. And to me, it wasn't an issue of like, or a question of like, is this going to work? It was more about, how do you actually make it work by pulling all these different services being delivered by different ministries or different verticals together? But we pulled it off. It was a partnership between academia, the government of India, several non-government organizations, communities. But it really was the NIH funding that started all this work and made it possible, with some support from the Elton John AIDS Foundation. So over the course of 4 years, we provided services to about 22,000 people at these 11 clinics we'd established. And as part of the research program, we had recruited about 47,000 participants. So that's almost 70,000 people who received an HIV test who otherwise likely would not have because of this trial. We showed that the overall model improved HIV testing by about 31%, which has since been adopted by the government of India. Now there are 71 such centers across India delivering these services for people who inject drugs. But perhaps the most important thing to come out of this model is it's been replicated for people who inject drugs in multiple parts of the world, including the US. And integrated services for HIV prevention and treatment are pretty much now standard for any population anywhere in the world, again highlighting that what we do in one part of the world has relevance in another. But integrating services was just the first step to meet people, and we knew this was never going to be enough. We've tried other interventions over the years, like, for example, can we add incentives to these integrated care centers? What if we integrated hepatitis C care? What if we started people on treatment the same day that they got diagnosed with HIV? What if we moved these services to the community? And with each of these interventions, we moved that needle a little bit. We were able to test and treat thousands, but we still saw a lot of new infections. So if you look at this figure here, you'll see the number of new HIV infections in the 3 largest prevention or HIV PrEP trials, which I'm sure Dr. Landovitz will talk about next, in the past decade. None of these trials included people who inject drugs. And at the same time, these 2 trials, 083 and Purpose 1, were running in sub-Saharan Africa to prevent HIV infection among people who inject drugs, among cisgender women with heterosexual transmission. People who inject drugs in the exact same region were almost 5 times more likely to pick up HIV infection, the exact same time period. And if you compare that to Delhi, 782 people in New Delhi had more HIV infections than the 12,000 participants enrolled across all these trials. And yet, there were no PrEP trials among people who inject drugs. Fortunately, as part of the HPTN and with support from NIH and Gilead Sciences, we are now actually implementing the first trial of PrEP among people who actively inject drugs. We are doing it in the US as opposed to India or South Africa, where the burden is significantly higher. But if we can bring findings from Bombay all the way to Boston, we might just as well be able to take our findings from Detroit all the way to New Delhi. But while designing this PrEP study, I actually ended up spending a lot of time with the communities in Baltimore, taking me back. So sort of like it's the full circle. I went from Baltimore to Chennai and now back to Baltimore. And in some ways, the lesson I learned was very simple and something that I'd known all along. So HIV, like most other infections, whether it's diabetes or hypertension, is very low in terms of the priorities in the life of someone who uses drugs. So they are people like you and me. Food, clothing, where am I going to sleep tonight? Harm reduction. That really was the priority. And somewhere down the line was HIV, which actually took me back to a day I spent in the field in New Delhi. So New Delhi, as part of one of our NIH-funded studies, we actually were trying to figure out what predicted HIV. Why was it so high in New Delhi? So instead of using our traditional approaches, we actually used machine learning and advanced data science techniques and AI to try and understand what was driving transmission. And contrary to what we thought in terms of individuals or people whom you injected with, there was one location, that purple dot, which was the strongest predictor of transmission. And I wanted to know what was driving this risk. So I went to this place. It was a blue shed, but behind the blue shed was essentially this open air drug market with thousands of people coming in and out. And I asked them, like, there is an ART center and a syringe service program literally 200 meters from you. Why don't you use it? And they look at me and said, when I don't know where I'm going to eat or where I'm going to sleep tonight, do you think I really care about my HIV? So I was actually with one of my friends from Abbott at the time. And so I turned to him and I said, what if we turn this blue shed into a clinic? And that really was the birth of the blue shed model, which married diagnostics and clinical care with basic necessities, teaching me possibly the best lesson that I've learned over the past 20 years. When basic necessities are met, that is when healthcare can begin. Just to show you what this partnership looks like in real life. So these were all the clinical services that we had, and we brought in haircuts, food, and showers and toilet. In addition to Abbott, we also had the Delhi State AIDS Control Society, the National AIDS Control Organizations, multiple community partners, but most importantly, the people who used drugs, they told us what services they wanted. And since we launched in November 2023, we provided service to almost 1,300 clients, the majority of who came for a haircut or a food packet, not for an HIV test. But when they were there for their food packet, they said, I might as well get tested for HIV. So we've diagnosed hundreds of people, started treatment for many of these people, and all our initial program data seems to suggest this model is working. We are currently actively seeking NIH funding to really use rigorous methods to evaluate this model, so this could be replicated everywhere else in the world. Because every time I have presented this model, whether it's in North America, whether it's in the US or in Canada, the first question I almost always get is, why don't we have this in the US? So I want to just end by saying this, like, yes, it is just a blue shed in one city in India. But the impact of these findings from this blue shed will have relevance to people who inject drugs across the world, including North America, because global drugs, global diseases is global solutions. Because in the end of the day, when you put the culture and the language and the race aside, people are people, regardless of where you are in the world. And in the US especially, which is a country of immigrants, and I'm one of them, right? So people are people. I want to end by just acknowledging first, and probably most importantly, the people who inject drugs who graciously participate in research studies globally. Our knowledge is advanced only because of them. I also want to thank my long-term collaborators in the US and India who have really put up with my craziness and all my crazy ideas for almost 2 decades now. And most importantly, the government of India and the community-based organizations that have been our biggest supporters. We've had a lot of funding sources, but I must say that none of this would have been possible if not for the support of the US National Institutes of Health. Not only did we impact lives in India, but we impacted lives globally. And for us to continue to make progress towards ending the HIV epidemic, all the way from Kolkata to Kansas, and putting people's lives first, we definitely need this continued engagement from the NIH. Thank you.
Thank you so much, Dr. Solomon. That was fantastic. That was just such an excellent talk, not only looking at the interrelationships with HIV and STI, also getting into the importance of really addressing those basic necessities and the interconnections that we have throughout the world. I couldn't agree with you more. That is just so critical to note right now, that it really, the US is, all of our countries are really intricately interconnected. And also from an immigrant family myself, so really appreciate sharing that. Really fantastic, and some beautiful comments here. I would see maybe we have time for 1 or 2 questions or comments from the viewers' audience. We've got one here shared for you. Some people had voiced some concerns about injectable PrEP for people who inject drugs. What are you thinking? What are your thoughts from your work with community?
So far, I think. Sorry, am I? Oh, sorry. No. So far, I think we're currently doing the trial in the US, so we can't really talk about the trial. But yeah, we're doing a good job. I would say, like, there are concerns definitely in the community, and I think the one thing for us to remember is there's no one silver bullet. You have a long-acting injectable, but there's also a long-acting pill that's in development, which I think could be more attractive to certain populations. It's also important to remember that people who inject drugs are different because we almost are seeing them all the time. So even weekly oral medications may be a good option because you're coming in for your methadone every day. So how hard is it to dose me? Dosing every day becomes challenging. I think it's important for us to think holistically and just remember that options are great. And longer is not necessarily better. That's the one thing I would lay out there. And it's not necessarily only injectables. We should be looking at the entire toolbox.
Yeah, fantastic. Thank you so much for joining and for this excellent talk. Really appreciate it. I'm now going to welcome in our next speaker, and really excited to hear from Dr. Raphael Landovitz, professor of medicine coming from another University of California, UCLA, going to be speaking on pre-exposure prophylaxis, past, present, and future. Thank you so much, Dr. Landovitz. Welcome. Oh, your sound. Thank you.
Okay, can you hear me now?
Yes, you sound great, loud and clear.
Great, thank you.
you.
It's only how many months since the beginning of pandemic, and we're still learning that we need to unmute on Zoom calls. My apologies. What I was saying before you could hear me is thank you again for this incredible honor of being invited to be a part of this amazing event, and thanks to everyone all over the world who's joining. I'm hoping to share today just some thoughts on the evolution of a specific type of biomedical HIV prevention that really has been brought to bear by an incredible longitudinal partnership between the NIH, other global funders, foundations, and the pharmaceutical industry to really transform HIV prevention since the early 2010s. And it's really been remarkable, but there's still a tremendous amount of work to do. Following Dr. Celum and Dr. Solomon is a huge set of shoes to fill. So I hope I can provide information, context, and be a little bit entertaining. These are my disclosures. It's not a surprise to anyone on this call that what we currently have in our prevention toolbox has not ended the HIV epidemic globally. And the numbers of new diagnoses of HIV globally still are at unacceptable levels. So we clearly have an enormous amount still to do in the prevention space. And we're going to be talking about a powerful intervention called pre-exposure prophylaxis that many of you are familiar with. And hopefully we can talk in a little bit of a nuanced way about where we are with it. So I don't want us to forget, as we focus on PrEP and biomedical prevention, that there's already an armamentarium, a toolbox, a menu of data-driven, evidence-based HIV prevention options. I'm going to focus today, because I was asked to do so, on pre-exposure prophylaxis, or PrEP. And a lot of people say to me, how did we get here? Whose idea was PrEP in the first place? Many of you are familiar with PrEP. Some may not be. So I hope you'll bear with me as we sort of get everyone onto the same page. Early on in the HIV epidemic, healthcare workers who might have gotten a splash or a needle stick with blood or body fluids from people living with HIV, and this was before we had treatment or prevention that was durable and effective for HIV, was really pushed the field forward with some observational work that was published by the CDC after animal models supported it, that after that kind of exposure, either a needle stick or a splash of body fluids from somebody who was living with HIV, the administration of anti-HIV medications as quickly as possible after that exposure could reduce the chance of HIV acquisition in the person so exposed. And what the observation was made fairly early on, that the timing from that event, the exposure to the administration of the medications, was one of the most important predictors of whether or not you were able to prevent someone from acquiring HIV from that exposure. The risk of such an exposure was not extremely high, but obviously when we didn't have adequate treatments, everything that could be brought to bear to prevent acquisition after such an exposure was really important. So if you follow with me and you agree that the evidence that, again, I'm going to ask you to go with me on this journey, suggests that time from exposure to medication administration was a critical parameter in the efficacy of this intervention, how do you minimize that time from the exposure to the medication administration? Well, you have the medication in the body before the exposure actually happens. And so that was the concept. It's actually sort of from calculus, right? How do you reduce the limit of time from exposure to dose to the minimal amount? Answer, you have the medication on board. I'm sorry, that was a really nerdy thing to say. I hope you all will forgive me. So that was the origin of the idea of pre-exposure prophylaxis. Anybody who has heard me speak on this topic know that I have a love of a certain conceptualization about the clinical trials that ultimately led to the first approved agent for PrEP. And the first agent to be studied was a combination of two potent and safe antiretroviral medications, tenofovir disoproxil fumarate and emtricitabine, we'll call it TDF-FTC. And it was studied in a variety of populations and contexts. This is my favorite slide on this topic. Those of you who've heard me talk on this before know I adore this slide since 2015, because I think it gives us an idea very early on one of the reasons that there was tepid enthusiasm about this intervention, about PrEP. If you look across the top row of the figures here, and the conceit here is that the shape of the figure represents the sex at birth of the population studied, and how filled up the figure is in blue represents the point estimate of HIV reduction in the clinical trial that's being described. And what you'll notice if you look across that top line is there was a wide variety of outcomes. Some were good, some were bad, some were in the middle. And if you sort of look on the leftish side of the slide, you'll see that it looks like the figures that represent individuals assigned male at birth look like they're generally more filled in blue than the individuals that represent females. And so this raised these questions of, does it work better in men? Does it work not as well in women? Why is that? Are there biologic differences between men and women? All sorts of such questions. And of course, the devil's in the details. And now we know that there are multiple considerations that contributed to these different clinical trial efficacy results. And some of it, probably the majority of it, has to do with adherence. If you don't take a medication, it can't work. And it taught us a really important lesson, which is for every new prevention strategy that is based on a molecule, so a biomedical intervention, we have to think about where the exposure to HIV in the body is occurring and whether or not the medications get to that location, or if in fact that's not even the relevant question. How well does it get into certain blood cells where early infection is established? So anyway, I'm not going to spend a lot of time on this slide, but you can see that there's a wide variety of results in large clinical trials that suggested that it probably could work, but we didn't fully understand the results. Happily, we have very smart people thinking about this. And if you go back and you look at people who had biomarker evidence of taking the medications at certain levels of adherence, you can actually tease this out a little bit from these large trials. And the short answer shown on this slide is, if you get the medications into the body with any reasonable fidelity, they're highly effective at reducing HIV acquisition in the people who are taking them. We used to have this conception that you needed near perfect adherence for vaginal protection. We now understand that that's not true. And, you can have some forgiveness for missed doses across populations and still have relatively high level protection. However, it is certainly a truism that the more reliably people can take these medications, the better and more robust is the protection. So this was game changing, right? This was proof of principle. This was really exciting. Of course, we come back to this idea, though, remember those clinical trial results. If you don't take a medication, it's not going to work. And there are all sorts of reasons: social and structural barriers to healthcare, stigma, side effects, racism, sexism, transphobia, so many reasons why people might not be able to take a medication like this with regularity. So a really smart guy in Paris, Jean-Michel Molina, made some observations that were rooted in some animal models, but also based on some really important community consultation that not everybody wanted to take a pill every day. And a lot of people, particularly gay men and trans women in the European context, were going out on weekends. And if they were able to plan that they were likely to have sexual activity, well, could you sandwich that planned sexual activity with dosing and get the same kind of efficacy we were seeing with more regular dosing in those original trials? And a lot of us who are providers said, don't do that. That's a bad idea. And of course, Jean-Michel Molina, way smarter than me, way smarter than a lot of us, said, well, let's study it. Let's study it rigorously. And he did a study called Hypergay in this incredible partnership with communities in France and Canada, and showed that if you sandwich planned sexual activity with a double dose of this TDF-FTC product 2 to 24 hours before the planned dosing, and then another single dose 24 hours later, and then a third and final single dose 24 hours after that, you could get incredibly high levels of reduction in HIV acquisition. And this kind of blew our concept out of the water, right? Again, we have to remember that the communities often point us to what they want, what they need, and it's up to us to figure out how to evaluate products that could be used in this way and how to create product profiles that are going to meet community needs. And Dr. Solomon referenced this in his talk. Dr. Celum mentioned it in hers as well. Understanding what communities want and giving them options is the best way that we are going to get people to uptake these sort of prevention interventions. So we'll come back to that. So some people said, well, but which one works better? And, I think it's unlikely we're ever going to see a randomized trial of daily versus on-demand dosing. But there was an open label extension to that IPER-GAY study called Prevenir. And basically what they found when they offered people either the option of taking the TDF-FTC on a daily basis or in this on-demand pericoital way that I was mentioning, the HIV incidence was really low and the same in both arms. And they averted an incredibly large number of HIV infections. And I apologize, that's a typo on this slide. It is not 361 HIV infections averted. It's a smaller number, but I apologize for that. So, that's all great. I do want to footnote here that the on-demand dosing, this 2-1-1, if you will, dosing, was only studied in cis men and trans women. And there's an evolving interest about whether this can work for vaginal exposures as well. And there's been some really elegant pharmacokinetic modeling that suggests that maybe, yes, it can be. And in fact, the United Kingdom updated PrEP guidelines actually does provide recommendations for an on-demand regimen for vaginal exposures. They call it 2+7. So it's a double dose and then 6 additional doses thereafter. None of these have clinical data to back them up, but we're hoping to fix that. So let's move on to the next product. So in the HIV therapeutic space, we know that TAF-FTC, which is sister or brother or son or daughter of TDF-FTC, it's basically like a different flavor of ice cream cone of the same thing. You like ice cream, you're going to like ice cream. But in the treatment space, it's safer from a bone and renal standpoint, although it does have other liabilities. It can increase your LDL cholesterol, can be associated with weight gain, but it is safer from a bone and renal standpoint. And the question was, could it work just as well, but be safer for PrEP? And so a head-to-head study was done called the Discover trial, that looked at this, and the long story short is it was non-inferior. So the answer is it worked just as well. We've stopped short of saying that it's safer. It's really a balance. This is a slide that I adore, created by Julia Marcus, formerly of the Kaiser Foundation, now at Harvard, that goes over this decisional balance, at least when both products are available and somebody wants an oral product, talking about the different things that you need to consider. I do want to say TAF-FTC was studied in cis men and trans women also, and got FDA approval in the US without any data for vaginal protection. There was a recent abstract at CROI last year presented. It was a post hoc analysis of a different trial I'll tell you about in 3 seconds, that did suggest high levels of protective efficacy with an average of 2 or more doses per week. We don't have more granular information, but the question is it time for guidelines to evolve to say that it's okay to use TAF-FTC on a daily basis for vaginal protection? My answer to that question is yes, I do think we're there. Is it going to lead to a regulatory approval? I kind of doubt it. Let's move to injectables really quickly. I've already used too much of my time getting us to this point. I hope you'll forgive me. The first injectable product, long-acting injectable cabotegravir, administered as a gluteal injection every 2 months after some starter doses, approved in numerous countries, but had lots of challenges with rollout. We could spend an entire talk unto itself talking about the challenges with implementation and how to get this to people. The clinical trials that led to the approvals shown here. Dr. Solomon already referenced them, HPTN 083 in cis men and trans women, and HPTN 084 in cisgender women in Southern and Eastern Africa, between a 67 and 90% reduction in HIV incidence in the injectable product compared to daily oral TDF-FTC. My read of these data, not saying anything about the individual potency of the product, but the injectable product covered sex acts better than a daily product that relied on a consumer-initiated activity to get that level of protection. So where are we with the products to that point? This is something that I stole from the IAS USA guidelines about, you have a person in front of you who might be interested in a PrEP product. What are your options? So summarized here, you'll notice that I put a question mark check for receptive vaginal or neovaginal sex for TAF-FTC. I think we're there. Most guidelines have not gotten there yet. And I'm going to sort of close with some brief comments about the newest kid on the block, which is generating a tremendous amount of excitement. Lenacapavir administered as an injectable product subcutaneously every 6 months, recent US and European regulatory approvals, and lots more approvals pending. And we're learning a lot now about rollout, which I think is really, really exciting. These are, again, the clinical trials that led to those regulatory approvals. These were a little bit of an interesting trial design. They were not direct comparisons. They were the active product compared to a counterfactual background incidence, which was a really elegant statistical analysis that led to about 100% reduction in cisgender women and 96% HIV reduction in cisgender men, transgender persons, and non-binary individuals. And I think it's going to be really exciting to see what happens in implementation. I'm just going to close by reminding you something that Dr. Celum and Dr. Solomon already mentioned, that we've got a lot of really exciting products in development. This isn't the end of PrEP. We're going to have vaginal and rectal inserts, microneedle array patches, hopefully a monthly pill, douche products, which we're really excited about, hopefully BNABs, and hopefully a vaccine, and even longer acting agents. This is a timeline that I've stolen from AVAC about when some of those products, including some combination pills with contraception, might be available. And I've already exceeded my time to tell you more, so happy to answer a few questions, and I'll thank you for your attention.
Well, surprise, it's me, Raphael. It's Judy here.
Hi, Judy.
Thank you. Sophia had to go. She's in the middle of a conference and the room was being overtaken. So thanks to Sophia for your excellent moderating, and welcome back to me, I guess. And I have had my wardrobe change, which I warned people I might do. Thanks so much for your presentation, Raphael. Fabulous as always. And I am just catching up to see what kind of comments we have. I think, let's see, 3:02, our time. I think we're okay to take a question or two. So we have one. Oh, I was just about to read that. In the world where the cost of injectable PrEP, due to patents in particular, seems to be so prohibitive. Can you see these questions? I don't need to read them out loud.
I can, yeah.
Okay, unless there are people who are not actually able to view, so we should read them out loud. So back to that, in a world where the cost of injectable PrEP seems to be so prohibitive that will not be available for so many people, how do we proceed? Advocate for cheaper drug pricing or write off injectable PrEP, at least in the global north, for the foreseeable future? Excuse me.
Yeah, no, thanks. I think it's an incredibly important question, and I think you've answered your own question, because this needs to be a call to action, as many of you have been doing for years, that we have to demand generics and equitable pricing, because it is not okay for these incredibly powerful interventions to not be available where they're needed most to the populations who need them the most. And there are many things in our world at the moment that are unacceptable and intolerable. Let's add this to the list and galvanize ourselves to advocate for this.
Thank you very much. I think in the interest of time, we're going to have to cut this off at this moment, but hopefully you can stick around and look at some of the comments in the chat. And I think what we'd like to do next, thank you again, really for excellent presentation. You're always so good and clear about and doing really important research, Rafi. So how about we play a video real quickly and then we'll move on to our next session.